Staphylococcus aureus is a gram-positive bacteria that produces a wide range of virulence factors that promote pathogenesis. These include bicomponent leukotoxins that disrupt the membrane integrity of polymorphonuclear leukocytes, also called PMNs or neutrophils. This video illustrates the isolation of PMNs from human whole blood and two distinct assays for quantifying S. aureus cytotoxicity against these important innate immune cells. These assays can all be accomplished with standard laboratory equipment and are easily adaptable to examine other aspects of host-pathogen interaction. These experiments will use whole blood drawn from human donors and will require approval from the appropriate institutional committee or review board, as well as a statement that informed consent was obtained from all participants. The first step of this procedure is the isolation of neutrophils through the use of centrifugation and density gradients. First, bring 50 milliliters of 3% dextran 0.9% sodium chloride, 35 milliliters of 0.9% sodium chloride, 20 milliliters of 1.8% sodium chloride, 12 milliliters of 1.077 grams per milliliter Ficol solution, and 20 milliliters of water to room temperature. Transfer 25 milliliters of freshly drawn heparinized whole human blood into two conical centrifuge tubes. Combine 25 milliliters of freshly drawn heparinized whole blood with 25 milliliters equivalent volume of room temperature, 3% dextran, 0.9% sodium chloride in a 1:1 ratio. Mix by gently rocking each 50 milliliter conical tube and then let stand at room temperature for 30 minutes. After incubation at room temperature, two separate layers will appear. Transfer the top layer of each dextran blood mixture into new 50 milliliter conical tubes. Centrifuge at 450 G for 10 minutes at room temperature with low or no breaks. Carefully aspirate both supernatant and discard without disturbing the cell pellets. Gently resuspend each cell pellet in 2 milliliters of 0.9% sodium chloride.